Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, single-cell chromatin proteomics to investigate the biology of exceptional longevity. Please join me in welcoming Dr. Simone Sidoli, our leading expert in mass spectrometry. In 2014, he joined the Epigenetics Institute at the University of Pennsylvania, where he developed pipelines linking protein phosphorylisation to chromatin changes. Since joining Albert Einstein's College of Medicine in 2019, he leads his own lab and serves as the scientific director of the Proteomics Core, advancing MS in disease and chromatin biology research. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further ado, I will now hand over to Dr. Sidoli to begin his presentation.
Okay, thank you. Thank you so much for being here and listening to my presentation. I hope I'll make it interesting. It's my pleasure to present an update of the work we've been doing in our lab at the Albert Einstein College of Medicine in New York. Specifically, we focus on proteomics to investigate exceptional longevity. I'm going to give you a quick introduction of what is the main mission of our lab. We develop mostly technology based on mass spectrometry to study the proteins that are attached to the chromatin in cells that undergo loss of homeostasis, which what we mean by that, we focus on this DNA that is being decondensing in specific cell states. And in particular, we're going to focus on the aging process. So aging is an interesting field of science because, like every science, it doesn't have, very specific starting date, but it has a very important milestone that is a sort of our turning point for aging science. And that is December 2nd, 1993, when this paper is being published in Nature. And it's a paper that basically presents a study on C. elegans, on the worm, where it shows that if you introduce a mutation into one of the worm genes, which is the gene DAF2, what happens is that the worm starts to live much, much longer, basically twice as long. This metabolism of this organism was like reprogrammed. It regulates lifespan to now basically face this longer period of life. So obviously this got aging scientists very excited because they immediately start looking in the human genome, where do we have the gene DAF2? So if we introduce a mutation, maybe we can live much longer. Spoiler alert, humans don't have the gene DAF2. So instead, we've been starting like a big quest for finding where is the secret of, eternal life. As you know, now there are many companies and academic labs that they work on extending lifespan and health span. This is just a list that I downloaded last year. I'm sure it's not updated, but there is a very high interest. And the main reason why there is so much interest is not so much because we want to live forever, but because aging is so correlated with a lot of human condition, nearly every human condition. So the graph that you see there on the left, that one is on a log scale. So the older we are, the more we are subjected to cancer, stroke, mental conditions and diabetes. So aging research is not about finding the cure for eternal life. It's about identifying mechanisms to basically reduce the incidence of basically nearly all of these diseases. And it was very interesting when I approached this field to find out that the answer to the question, what causes aging at the molecular level, is still not fully answered. So we as a lab, we are working with many others to try to understand the mechanism that trigger the aging process. And I could speak for hours because there are of course lots of labs that do amazing work on this field. But among all the hallmarks of aging, probably the one that has more like general consensus of where things start is towards DNA damage, and specifically DNA mutations. So during the aging process, we accumulate mutations in our DNA, insertion, deletions, point mutations. This is one graph from a paper of a close collaborator of ours, but they could show many others. And the thing is that, of course, these mutations accumulate with time. The issue is that these mutations seem to be of stochastic nature, so they don't seem to have very specific patterns, and especially there's not very specific mutation that correspond to specific extended lifespan. So in our lab, we try to focus on not just the mutation, but how this phenotype changes. Because one additional problem to this DNA stochastic mutation is that they are not only different between you and me, they're also different within our cell population. So within our cell population, the accumulation of mutations are different between different cells. And in fact, if we create model of aging in the lab, for example, by inducing radiation, so what happens is that when we do single cell proteomics, we notice that the young cells are not only different from the old cells, but the old cells are different from one another. So you see that there is so much more variation among cells that have been irradiated to model DNA damage and aging. So where we think is the answer, we think is not in the mutations of the DNA, but on the structure of the chromatin itself. So chromatin normally is depicted with cartoons like this. There are regions of chromatins that are more compacted, where DNA is less accessible, where there is no transcription, where there are usually DNA methylation, and in particular, there are certain type of histone modifications. And then other part of chromatin instead are decondensed, and here is where we have transcription and other type of histone modification that promote the recruitment of transcription factors. Usually, histone modification are on these tails that protrude outside the nucleosomes with this assembly of histones, DNA wrapped around it, and of course, they have a lot of important biological function. If you Google histone modifications and functions, usually you get a table like this, maybe now a little more populated, but of this general size, and you see that there's a handful of histone modifications that are associated with active transcription, basically promoting open chromatin and RNA production. And then there are a couple instead that are associated to gene repression. Either regions of constitutive heterochromatin that are basically locked up for almost the entire life of the cell, or regions of facultative heterochromatin, where chromatin is a little more dynamic, but nevertheless usually silenced. Truth to be told, chromatin, now we know more than 500 different type of histone modification, also thanks to mass spectrometry, mostly thanks to mass spectrometry. But there are so many that for which we don't have a very clear biological function known. And the most important issue that I want to highlight is that during the aging process, chromatin starts to assume like different shapes, like it decondenses. There are regions of chromatin that are still decorated with certain type of modification, but now they are more accessible. There are many reasons for that. Not only mutations contribute to loss of homeostasis of the chromatin, but also we lose nuclear lamin, the protein that is on the surface of the nucleus. We lose DNA methylation with certain type of heterogeneity, and therefore we need to find out what happens in these decondensing regions of the chromatin, where there could be a whole new set of molecular mechanisms that define new function for this chromatin, and maybe preventing the decondensation or the mechanism occurring in these domains. That is what help us figure out maybe a way to extend the health span first of the cell and possibly the entire organism. So these are the questions that we try to address in our lab. How reproducible this is, like what are these histone modifications, what kind of proteins interact with these modifications. And we have a number of projects that are focused on this, each of them, of course, different lab members. It's kind of ambitious to go through all of them in a single presentation. I'll try my best to be not too complex. First of all, the first question we need to address is that how do we study these histone modification, and can we study the heterogeneity of these modifications? And actually, we do it with a regular proteomic workflow that is a little bit modified to focus on histone marks. For those that are expert of the field, this is obvious. For the others, just quick reminders that we extract histones or other proteins from nearly any tissue or cell. Histones, they undergo a slightly different sample preparation. We extract, let me see if I find the laser pointer. Here it is. We extract histones with an acid extraction, and then compared to the regular proteome, there is a step of derivatization that we do, a method that has been optimized by the lab of Ben Garcia. And now we analyze them with mass spec. So there's a few extra steps compared to the regular proteomics, but luckily we've been able to optimize them using the cell-in-one. So using this liquid handler that is optimized exactly to prepare single cell. We deposit single cells in well plates, and then we do this sample preparation within single cell. We have a paper in bioRxiv, hopefully will be published soon. Thanks also to our collaborators at the Broad Institute. Now basically we do single cell histone PTM analysis, and results are really striking. As you see from these chromatograms, we see a very clear signal to noise even when we inject a single cell into the mass spec, and a very linear response depending on how many cells are deposited per well. So this is remarkable for us because we're not talking about proteins, we're talking about protein modification, which are always substoichiometric. But now with the possibility of detecting those, we quantify between 50 and 60, we can assess heterogeneity within cell population. And actually, we also show examples where we can demonstrate that we can quantify whether all cells respond to certain treatment in the same way. It's a long story, but cutting to the chase, why this is relevant in this presentation is because by assessing this heterogeneity, we can quantify, normalizing by the technical variation, we can quantify biological variation of these histone PTMs. So this graph here on the left shows which histone peptides are the most variable cell by cell in the model system we are using. And what is interesting is that the ones that are at the top of this table are histones that are co-modified with modifications that are actually not supposed to coexist on the same histone. In particular, these two, lysine 9 trimethylation is a well-known marker of compacted heterochromatin, while acetylation is usually associated with open chromatin. So we have this histone mark that is relatively abundant and heavily heterogeneous across cells within a population. So we're wondering if we are starting to quantify this region of chromatin instability, right? That is not as reproducible between cells. It's it's a long shot. So there's a lot of things we need to do to actually prove that we are talking about this type of chromatin. The first step is we verified that this hybrid histone modification occupy regions of open chromatin, and we do that by sucrose gradient. So basically we cross-link and centrifuge chromatin. The compacted chromatin will form bigger complexes compared to the open chromatin that after sonication breaks into single nucleosomes. And then by separating it physically, we can run all of this fraction by mass spec and verify that in the late fraction, we have the histones modified with our heterochromatin markers, what the hybrid codes instead, they come out in the very early fraction. So at the very least, we know that they occupy regions of open chromatin. So one question we ask immediately is, what proteins recognize these strange hybrid modifications, because there could be proteins that are involved in reading these regions of decondensing heterochromatin. So I have a few slides about one of the projects we're running in the lab where we identified a protein that seems to be recognizing specifically the hybrid codes. And the protein is called XOSC4, which is a protein that is part of the exosome complex. I'll get in more detail in a second. First, I want to show you how we identify these proteins. We do them with experiments of peptide pull down. So we mobilize on beads long histone tails that are modified with silencing mark, acetyl marks, or the combination of marks, and then we look at which protein seems to be particularly enriched by the combination. And you see that this protein is one of the most enriched, very specific compared to the other controls. We also do the reverse experiments where we immobilize on beads XOC4, so the protein itself, and then we check which histone peptide, which histone, they bind to this protein. And you see that if the histone is co-modified with the hybrid code, it very much prefer to bind to these beads. So there is this mutual enrichment that we get. And XOC4, like I said, is part of the exosome complex. Is actually already known to be a protein that is annotated to have a critical role in eliminating non-coding transcripts. So it does make sense that it might recognize chromatin that is decondensing in a non-regular way. It's not an enzyme, it's a component exactly of this macromolecular structure. And we try to we're working on the crystal structure. We tried to model this with AlphaFold, and we actually see that the modified histone tails might have some point of contact with this protein. Why this is interesting? So we take our cell culture and we induce these regions of decondensing heterochromatin. We use some treatment with the sodium butyrate. We increase the relative amount of this hybrid code on the chromatin, and then we see that we actually have an enrichment of this protein bound to the chromatin, and therefore we think maybe this protein is getting there to protect from spurious for excessive transcription. So this is something that we find out by knocking down the protein. If we do a knockout, the cell is very sick, but if we do a knockdown, the cell is alive for a while. So what we can do, we can see what happens to the transcriptome. And then you see that there is this massive shift into the number of transcripts that are being produced by the cell when this protein is missing compared to our scramble control. If we do a gene ontology of these RNAs that are the most regulated, senescence is one of the markers that comes up the first. But we think actually the most interesting aspect of this volcano plot is not really what kind of genes are regulated, but the fact that the majority of these genes that are regulated are actually non-coding transcripts, so not classic genes. Here is an extra slide where we show that if we treat cells with sodium butyrate, so if we put an enormous amount of acetylations on the chromatin, we don't have the same regulation. So it's something that is very specific to our XOC4 knockdown. There's no correlation between removing this protein or having a wild type cells that is treated with this excessive amount of acetylation. And like I said, the most interesting part for us is the fact that most of these transcripts that are being over-represented are non-coding. And in fact, if we look at the protein coding transcript, there's not much difference between the knockdown and the control. So the issue that we have with this non-coding transcript is the fact that many of them we don't know yet. So one aspect that has been exciting for us looking through the data is the fact that quite a number of these non-coding transcripts that are upregulated, are more abundant in the knockdown cells, are antisense. antisense, potentially they have some post-transcriptional regulatory effect compared to the sense genes that they match. And in fact, if we plot them on the graph, these antisense that are more abundant in the knockdown, they correspond to basically the lower levels of the transcript corresponding to the respective genes, which is saying something because there's not a lot of genes on this left part of the graph. So that's that's what we think is happening. So now what we have in progress are experiments where we block this antisense and we see if we can restore the phenotype. This work is still unpublished, so I can only offer you temporary conclusion for now. Which is exactly, I hope I convinced you that now we have a method to look at histone modification in single cell, quantify their biological variation, and potentially investigate the partners, so the proteins interacting with this histone modification, which help us define a potential biological role of why they become more abundant on chromatin and why they are more heterogeneous. But now the problem is that these are cell models, right? So we wanted to check if any of these phenomena are reproducible in real, aging individuals. And so when we look at individuals, we have a new story to tell. And with this one, I go towards the end of my presentation, which is another histone modification that we think gets enriched in this during the aging process. So, like I said, it's a very exciting field of science because it really allows us to study a phenomenon that happens to all of us. And here at Einstein, we are lucky that we have access to a cohort of donors who are centenarians. This is a picture from PLOS Biology in 2006 that shows the same people many years apart. And some of these individuals have been donors at Einstein, and they basically give us their blood so we can purify T cells and B cells and look at whether there are secrets for exceptional longevity. Now, the problem of comparing individuals in their 50s with individuals that are 100 years old and older is that, of course, there are differences. We don't need a mass spectrometer to see that they don't have exactly the same phenotype, and therefore this creates confounding factors about what is there in their blood because they have exceptional longevity, or what is there in their blood just because they are very old. So what we have in this cohort, we also have the privilege to have access to the progeny of these centenarians, which is this group right here. So these people are basically the children of those that live 100 years and older. And so we can age match them with the controls, and then we can actually compare what is specific in this group compared to the others. Because statistically, we know that children of centenarians are also more likely to live exceptionally long life. So, of course, the first thing we did when we got access to this sample, we started looking at histone modification. Is that, do we see any regulation in this hybrid modification? Is there less of this hybrid modification in individuals that are projected to live exceptionally long life? And what we noticed that global levels, no huge changes in histone modification. When we focus on our specific hybrid modification, it was actually exciting to see the centenarians seem to have less of this mark. I forgot to write it here, sorry. These are B cells. There are 5 individuals per group. But overall, what we really wanted something that is specific for the progeny of centenarians, something that is very low or very high in this group that tells us that these people are more likely to live exceptional life in the future. We looked through all the histone modification. It was very hard to find some specific histone modification that look unique, at least among the classic modification that we usually screen for. So basically what we did, we just extracted the chromatin proteome of these cells, and we look at what kind of other proteins are present in these 4 groups to see if maybe we are missing something. So the chromatin proteome We identified around 3,500 proteins from the chromatin isolation of these 4 groups, and we clustered protein relative abundances between different trends, and we like to focus on this one, on the group of proteins that seem to be spiking, seems to be more abundant in the group of the projected exceptional longevity. What you see there on the right is the gene ontology of the proteins that belong to this group, using as a background control protein identified in the other groups. There is a lot of obvious terms like proteins involved in regulating RNA. Of course, there are chromatin proteins, but among this group, there were some proteins that they seem to be specific for a certain histone modification that we have been overlooking, which is histone succinylation, which we have not investigated at all. So succinylation is a modification that we normally don't screen in our histone analysis because it tends to be very low abundance. And the other thing is that I don't want to offend anybody, but literal tool so far has shown that it's not as interesting because it has a molecular structure that is different from acetylation, but not as much. And in particular, there are these studies where they do chip sequencing, where they map these modification on chromatin. And the conclusion is that they somehow occupy more or less the same chromatin domains occupied by acetylation. So some of the general conclusion is that it might be just acetylation in disguise, just much less abundant, but with similar function. We are not so convinced about that, and I'm going to show you why in a few slides. First of all, the reason because succinylation is interesting in the aging field is that succinylation comes from the precursor succinyl-CoA, which is an intermediate of the TCA cycle. And succinyl-CoA comes right after alpha-ketoglutarate. And alpha-ketoglutarate is a metabolite that, if given to or mouse models, they actually extend the lifespan and health span. There's a number of studies. Sorry, I forgot to cite them, but there's a number of studies where they show this. And this was already interesting per se. Like, the other interesting thing is that by looking into our data, we have these spectra produced by our canonical histone analysis by mass spec. Now we started to extract the succinylated histone peptides, and indeed we can see that there is higher level of histone succinylation in B cells and in T cells in the group of individuals that is the progeny of the exceptional longevity. There is higher levels in B cells. In the T cells, we only compare this group with the age match control, higher level of certain histone succinylation, alpha-ketoglutarate, succinate, also the metabolites that are associated to that. It was exciting for us because it got us thinking, well, there is more succinylation in these individuals that are projected to live longer. Does it have any meaningful role? So the first thing we did is that can we model this in a cell lab so we can focus on the biochemistry? And the first interesting thing we notice is that, yes, we can increase levels of histone succinylation by adding succinic acid to the cell culture. Error bars are wide because modification is low abundant. But more interesting than that, we noticed that we can boost levels of succinylation even if we add drugs like metformin, which are known to be under the spotlight as a potential gerotherapeutic. So basically, drugs that extend lifespan even in human. There is more succinylation if we add these molecules. There is more succinyl-CoA specifically in the nucleus, not overall in the cell. And this analysis is possible thanks to our collaborator Nate Snyder at Temple, who can really perform like nuclear metabolomics, like we can focus on the metabolites inside the nucleus. So at this point, we were wondering why succinylation is enriched in this group of individuals who are projected to have exceptional longevity, either by substituting acetylation it might prevent spurious transcription of chromatin that is anomalously decondensing, or maybe succinylation actually has some binding partners, some proteins that are recognizing this mark, and these modifications, they maybe contribute to maintain compact chromatin, silence transcription, let's say they maintain the chromatin younger somehow. Okay, so the first experiment we did, of course, again, we took our peptides, we modified them with succinylation, specifically on the residue 64 of histone H3, which was the most abundant we detected in our mass spec analysis. And then with an acetylated peptide as a control, we started to look, is there anything specifically enriched by succinylation if we use as an input material a nuclear proteome? Interestingly, you see that these volcano plots are quite skewed to the left. So there are many more proteins recruited by acetylation than recruited by succinylation. That maybe is not too surprising because we know that acetylation has a lot of binding partner, a lot of transcription factors. In fact, if we zoom in into this volcano plot, we can see that all of these proteins with bromodomains that are associated to transcription, they're all, of course, on the left side of the volcano plot. They prefer acetylation. So this one is actually interesting per se because it helps us potentially address the first hypothesis we had. Maybe the accumulation of succinylation prevents transcription, right? So there's something, I'll tell you immediately that that's an answer we don't have yet, but it could be interesting because it could prevent proteins with bromodomain being excessively recruited on the chromatin of decondensing chromatin. But we also noticed that there are a few proteins that instead prefer to bind to succinylation, and those seems to be proteins that are lysine demethylases. So enzymes that are in charge of removing methylation from proteins. And then what happens is that when we focus on these lysine demethylases, and we did it with the collaborator in our department, JP Mayanti at Einstein, what we notice is that it does make sense that lysine demethylases might be recruited by succinylation. Because if you look at the active site of lysine demethylases, which is depicted here in the graph, if we substitute the methylated histone in the active site with the model of a succinylated histone, what happens is that it seems to fit and satisfy basically the coordination sphere of this iron atom, and basically get stuck into the lysine demethylase. So this one could have both the effect of recruitment and maybe the effect of silencing, basically inhibiting the activity of this enzyme. So once we predicted this, what we did, we tried to do some enzymatic assay, and we actually looked whether any KDM gets inhibited by the presence of a succinylated peptide. What you see in this graph, here you see a positive control. Basically, this is an inhibitor of lysine demethylases that around 1 micromolar, so this is not molar, this is micromolar. There is an inhibitor effect, and then this is a peptide that inhibits a bit, not much. You need a really large amount. But if you try on an array of lysine demethylases, there are a few where this effect is more pronounced. In particular, these two, lysine demethylase 4D and 6B, which are not two random KDMs. Actually, they are maybe the most interesting in the panel that we investigated, because those seem to be the ones that are most inhibited by the presence of succinylation. And these two enzymes are actually the ones that remove the silencing marks. So 4D is also involved in removing lysine 9 trimethylation from histone H3, and 6B lysine 27 diene trimethylation, which are the two silencing marks that I mentioned at the beginning of my presentation. This was really exciting for us. So we immediately checked if we induce succinylation on the cell, if there is, let's say, higher levels of these silencing marks, and that's exactly what we see. So maybe succinylation is also involved in basically reducing the activity of lysine demethylases, which normally remove silencing markers from heterochromatin. This is where we are until now. Now we are doing transcriptomic experiments, ChIP-seq experiment to map all of these phenomena on the chromatin and understand what is their impact on transcription. But for now, I'm going to stop here. And these are the experiments that we have in progress, which I hope I'll be able to update you with that very soon. And of course, thank you to all of our collaborators. My conclusion, I hope I convince you that studying aging with mass spectrometry is very exciting, especially when you focus on the chromatin proteome, where there are a lot of mechanisms happening as our chromatin decondenses during aging. These are all the people I want to thank. There are many more. Sorry if I forgot some, but thank you to the lab, thank you to the funding, thank you to the collaborator, and thank you, to all of you for your attention.
Thank you very much, Dr. Sidoli. As we go through the Q&A portion, I encourage all of you to participate by submitting your questions in the chat now, and I'll do my best to get these questions over to you, Dr. Sidoli, during the session. So to start us with, there's a question about centenarian progeny. Is there any difference in phenotype, habits, and health in progeny of centenarians compared to controls?
Yeah, thank you. Thank you for the question, and thanks again for the invite to speak at this event. Yeah, it's really interesting about this discussing about this aspect because centenarian have been studied for a long time here at Einstein in our institution. It's a program that began with Professor Nir Barzilai, that is actually acknowledged in the slide, and there is a number of epidemiologists that have studied the progeny of centenarians, and indeed, when they are age matched with controls, they have better metrics in terms of health, in terms of cognition, in terms of frailty, the lower frailty. So there is that variable that we need to take into account. So they are not. We're not only talking about chromatin. Actually, we don't know yet. What are the secrets of chromatin of progeny of centenarians, but indeed they are healthier than their counterpart. Do they live a different significantly different lifestyle? The answer is usually no. So there's no significant difference. Like, it's not that they eat healthier, it's not that they run more than their counterpart. So this is very important for our study, because that will introduce a bias into our model.
Thank you, Simone. Now moving into the molecular details, what do you think is the major chemical impact of succinylation on chromatin? How does this modification influence chromatin's function, particularly in relation to aging?
Yeah, that's a good question. So what succinylation does to aging, obviously the final answer, we don't know it. That's why we are working hard on this program. But in general, succinylation has been discovered now more than 10 years ago. There are not that many papers that focus on this modification. And I think part of it is because it's considered to be a little bit like acetylation in disguise, because it has a similar carboxylic group. So the difference with acetylation is that acetylation state of the lysine from +1 to 0. Succinylation moves it from +1 to -1 because it adds basically an acidic group. So there is a difference in the charge state. That one, in theory, should contribute to make chromatin even more condensed. And we don't know if it's true because we don't have this kind of structural data, but we do know, as I mentioned during the presentation, that the bromodomains, so that are typically associated with activation of transcription, they struggle in binding to this modification. So we think, to summarize, we think that this modification is indeed chemically contributing to decompress the chromatin, but not as well activating transcription like acetylation.
And continuing on post-translational modifications, Is there any other modification that inhibits lysine demethylases in vitro? Is succinylation unique in this regard, or are there other modification with similar inhibitory effects?
Yeah, thank you for this question. Actually, I didn't put it in the presentation, but we have done these experiments. So if we use FUMA-related peptides, we get inhibition of KDMs. If we use malonylated peptides, we get inhibition of KDMs. So it's not just succinylation. The reason because we focus on succinylation in our work is because we don't really have any evidence that histone fumarylation or histone malonylation are detectable, are present in detectable levels in the cell of these centenarians, in this progeny of centenarians. But indeed, from the chemical perspective, there are other post-translational modifications that inhibit KDM.
Thank you, Simone. Moving on to a different topic now. So, single cell analysis is such a powerful but challenging tool. We have a question asking how many modifications are detectable with single cell analysis? Could you speak to the current capabilities and limitation in this area?
Yeah. Thank you for that question. Yeah, of course. Obviously, when we prepare samples from one cell, we don't get the same sensitivity as bulk material. When we study bulk material now, we detect regularly more than 300 different combination of modifications, so acetylation, methylation, phosphorylation, and a little bit also the other like succinylation like we discussed. When we bring the material down to single cell level, so we're talking about 10 picogram of starting material, then we detect with confidence around 50, 60 different marks, which is still remarkable. Unfortunately, some of the very famous modification are below the detection level, lysine 4 trimethylation from histone H3, which is a very important marker of active promoters. It's a low abundance modification, so when we do single cell at the moment, we don't easily detect it. But we think that there is room for optimization there. The paper that now we have in bioRxiv and it's in revision, we actually show that we can optimize a little. There is still room for optimization in the sample to bring up the sensitivity. And of course, as mass spectrometers become more advanced, this will become easier and easier.
Thank you. I think you just cut off at the end there, but on a related note, can you just repeat if you can quantify differences in cell treatment at the single cell level, or this? I mean, this sounds incredibly valuable for understanding heterogeneity, but what does the process actually look like?
Yeah, exactly. So this one is something I quickly touched in the presentation because obviously I wanted to give enough time for everything. But what we are mostly excited about single cell is not really, of course, the flexing that we can analyze histones from one cell, but really what is the biological heterogeneity. And this includes both the fact that we can see within a cell population which histone marks are the most variable, so which probably benchmark regions of chromatin that is not very reproducible, is not very stable, but also and we have this in our manuscript, as we treat cells, we can even see if all cells respond to a certain treatment equally, which is something that, of course, when you do a blend of cells and you do a proteomic analysis, you cannot tell. We can really cluster cells that are treated with, in this case, HDAC inhibitors, and we can see which ones respond more or less. The reason because it can be done is because we have come up with a method to normalize to obtain true biological variability. So first we create a calibration where we test what is the technical variability for each peptide, and then we do it with a standard that we know is spiked in always at the same amount. And then we basically normalize the biological variability by the technical variability to get exactly the true biological variability. There's still a lot to be done, but we think this is a very powerful technique, for example, to study virus infection, for example, to study tumor microenvironment, right? Because within a microenvironment, not every cell will have the same phenotype, the same function. So yeah, we are looking forward to apply this to a lot of different questions.
Yeah, so let's just return to the topic of succinylation in aging again. Are there any known pathways or enzymes responsible for succinylation in age chromatin, and do these differ in longevity associated profiles?
Yeah, we get this question a lot. Think about succinylation. So the precursor is succinyl-CoA, right? So that's a molecule that is similar to acetyl-CoA. So acetyl transferases, they use acetyl-CoA, but they have quite a spurious activity in utilizing succinyl-CoA as well to modify proteins. So there's not really a well-defined list of enzymes that catalyze succinylation. They are mostly enzymes that catalyze acetylation as well. I should also mention that succinyl-CoA is a molecule that is reactive by nature, right? So if you actually put in an Eppendorf tube recombinant naked histones and succinyl-CoA, you get some succinylation because the molecule actually lysines are very reactive with succinyl-CoA. So part of it is chemical, part of it is enzymatic. Some publication, including one that I mentioned in the presentation, they boost level of succinylation by using inhibitors of HDACs. So histone deacetylases, if they are inhibited, they also contribute to increasing the level of succinylation, which means that also the erasers, some of them are the same enzymes that also remove acetylation. Sirtuin 5 and 7 are currently the best candidates to be the erasers of succinylation more specifically. But like I said, there is a lot of promiscuity.
Thank you, Simone. We actually have a bigger picture question here that have just come in. So, could widespread application of longevity-related chromatin modification interfere with natural selection processes? Should we be concerned about potential unintended evolutionary consequences?
That's a very interesting question. Yeah, the thing about aging science is that you need to take so many things into account. So it's not only about science, it's also about society, bureaucracy, philosophy, maybe even. And it's it's obviously I don't have a very good answer for that question. That is very interesting. Uh, what I can say, we receive funding. Actually, you can see it in this slide. We receive funding from international institutions because, to be honest, the reason because they put the government or even like Saudis, or, of course, the United States. The reason because they fund treatment to live longer.
I'm sorry, Simone. You just cut off just on that last sentence. Could you just repeat it? What you just said? Sorry.
Yeah, sorry about it. I was just saying that the reason because we get this funding is no, I don't think anybody expects that we find a cure to live significantly longer. They expect us to find the mechanism to extend the health span. So let's say we're going to live 100 years. Can we live most of these years in health? And that's why governments from many countries, they are interested in this because the problem is we can live as long as we want, we consume, we buy things, like also after we are retired. The problem how much individuals are going to wait on the health system, right? And obviously because healthy individuals, it's a benefit for everyone. So the reason because we study aging is not really to live longer, is to have a healthier lifespan. And regarding the evolution, I wish I had a better answer for that. I don't really know how to comment beside the fact that it brings up the question whether um, if we are going to be healthy until the last day of our life, will we be ready to go, right? So what is going to do to our society? Let's say if we really come up with the treatment to be healthy until the last day of our life, how this is going to impact society, and potentially, how how we are going to change. That is a question that I don't have a good answer, but I'm really glad you brought it up.
Yeah, it's definitely an interesting one. And we do have another, I suppose it's more closer to a controversial question for this final one. Some studies have suggested that pushing cells to avoid aging or death can lead to cellular senescence, which is kind of like linked to cancer and other diseases, I suppose. So, does your research consider or mitigate these potential adverse effects?
Yeah, senescence is of course a very big topic in science, something that we don't work on directly. A lot of our colleagues work on it. Senescent cells are sort of considered like zombie cells, right? Because they are alive, but they are, theoretically old, but they are not as functional as they used to be. Part of the actually a big branch of aging scientists, they study methods to basically get rid of the accumulation of senescent cells. And that one, from their perspective, is the mechanism to keep organism younger for a longer time. You can do it through autophagy. You can do it through exactly killing the cells. There are different mechanisms. We don't have any evidence that giving succinic acid to our cells induces senescence, but obviously when you study this in a more complex system is a variable that we need to consider.
Thank you so much, Simone, for your thoughtful and detailed answers. So we appreciate your time and insights into this fascinating field. And thank you to everyone in the audience for joining us and for your excellent questions. So, like all the talks from this event, this talk will now be available to view on demand for the next 30 days. Please do continue to submit any additional questions you may have, and this will all be sent to the speaker after the event. Once again, thank you for your attention, and a big thank you for Dr. Simone Sidoli for joining us today. And we will be back shortly with the next talk.
Thank you.